Thank you, Blakely, for this great introduction, housekeeping, and valuable information. I think all of you that are here already know that Leishmania is a hot topic for the last few years, and that's why we have such a good number of people today. And we also have a fantastic topic today and fantastic presenters, so we're gonna have a great 1 hour of being together. And I'm not gonna waste any more of your time, but I would like to present you our first speaker. His name is Dr. Dvořák. He's an assistant professor at Charles University in Prague. He specialized in vector biology, focusing on sandflies and their role in leishmaniasis transmission. His work spans in molecular taxonomy, ecology, and epidemiology, which makes him a first choice speaker for Validate Network. Vid, floor is yours. Please share your
All right.
presentation.
Thank you very much, Sarah and Blakely, for your very kind introduction. And good morning or afternoon to everyone, depending on your time zone. I'm extremely happy to be here today. It is an honor and pleasure at the same time to present, along with my two co-speakers, Iva Kolářová and Suzana Blesić, our project, CLIMOS project, that aims to focus on the fly-borne pathogens and potentially the development of some strategies how to raise awareness and how to help the public stakeholders to actually cope with the challenge of leishmaniasis and other fly-borne pathogens. So I will quickly start to share my presentation. Can you all see my screen, please?
Yeah.
Yes.
Excellent. Yes. Thank you very much. Okay, so as I said, it is my great pleasure to talk today about the CLIMOS project, which is a Horizon Europe project aiming to monitoring and decision support framework for fly-borne diseases detection and mitigation, as the long full name of the abbreviation CLIMOS says. We named our talk Sandflies, Tiny Insects, Important Challenges, and I think that indeed kind of reflects the, at one side, small body size of the insect, and on the other side, the epidemiological importance of sandflies as the vectors of important and challenging human infecting as well as animal infecting pathogens. I couldn't resist to include an introductory slide to show you how actually a sandfly looks like, which you can see on the left side of my slide, while the lovely insect shown on the teaser of our today's talk is actually not a sandfly, but it is a mosquito. And we are not offended by that by any means, of course. This is totally obvious because the mosquitoes and mosquito-borne diseases are of great importance, medically as well as veterinarily. So we're pretty much used to that. Just to give you an idea that sandflies indeed are sort of smaller cousins of the mosquitoes among the large group of the dipteran insects. Dipteran referring to the fact that they only bear one pair of wings for their flying. Nevertheless, sandflies are tinier in their body size. And also some of their other biological characteristics differ and differentiate them markedly from the mosquitoes. So while we, the sandfly people, heavily rely on our colleagues in the mosquito research, we quite often need to cope with different challenges and different traits of our research due to the fact that the sandfly biology is actually in many aspects quite different from the mosquito biology. Nevertheless, the importance of sandflies as the vectors definitely lays in the fact that they do transmit some medically important pathogens, and these are numerous. Nevertheless, for the European context, there are two major and most important groups of sandfly-borne pathogens, and these are leishmanias, which are protozoans, pathogenic protozoans that cause a number of differently manifested diseases in people and animals alike. The transmission can be mostly zoonotic, meaning that they have an animal reservoir, but also it can be anthroponotic, which is to a lesser extent in Europe. There is a number of different Leishmania species transmitted in Europe. All of them transmitted to people are nevertheless vectored by the sandflies. And there is another large group of pathogens that are also fly-borne pathogens, and these are phlebiviruses. Out of those, Toscana virus stands out as one of the most important one, causing symptoms in infected people. Nevertheless, the group of phlebiviruses is a broad one, and it contains a number of viruses that some of them have been only recently discovered, and their medical importance is nowadays under consideration and under investigation. If you look on the sandfly fauna in Europe, you can see that the number of sandfly species occurring in the European region is actually not overwhelming. It is a modest number of about 25 species, mostly concentrating in the Mediterranean countries. And we can conclude that all the Mediterranean countries, being it European Mediterranean or the North African Mediterranean, they are all endemic for a number of different sandfly species, most of them actually being capable of transmitting Leishmania of several species, as well as some of the phlebiviruses. However, we expect, and we already witness, a shift in the geographical occurrence of sandflies due to ongoing climatic and environmental changes, which has been reflected in field data that actually document the introduction or emergence of sandfly species into regions where they were previously not present, which is actually shown on the two small maps of north part of Italy, documenting the introduction of Phlebotomus neglectus, a particular species of the subgenus Larusius, into that region, where it was barely present in the 1990s. Nevertheless, in the upcoming decades, it was documented to be spreading into this area. And moreover, we also can model, based on the climatic variables, the probable emergence of sandflies into new geographical regions in the next decades due to the, as I was mentioning, ongoing climatic and environmental changes. And therefore, it appears crucial to actually understand these drivers of the emergence in order to be prepared for the emergence of the sandflies into the areas where they have not been present before. Interestingly, the sandfly fauna, even though quite moderate in the number of the species, is still not fully understood. If we consider that the first sandfly species has been described as early as 1786 by a prolific Italian naturalist, Scopoli, then it's quite surprising that in the last 25 years, we actually witnessed a number of new species being described from the European region, which could have been considered as thoroughly investigated. Nevertheless, still we come across newly described species. Phlebotomus economici, starting the 2000 decade by the description from the island of Cyprus, which has been the actual last description of a sandfly species solely relying on the morphological data. But in the upcoming years, a number of different species, Phlebotomus anatolicus and kiliki, Phlebotomus criticus, and finally Phlebotomus simahalepe, were described from various regions of the Mediterranean or non-Mediterranean Europe, actually combining different approaches of classical morphological taxonomy together with molecular techniques like DNA-derived techniques or multi-of protein profiling. So interestingly, we can still see new species being described from our very backyard, this only emphasizing the fact that our understanding of the epidemiology of sandfly-borne diseases is actually far from being complete. Moreover, as I was already pointing out, we can see more and more recalls of sandfly presence from regions and countries which are well off the usual Mediterranean area of sandfly distribution. And this is just an overview of recent publications indicating or demonstrating the presence of sandflies in different non-Mediterranean countries such as Switzerland, Belgium, and that is a very recent article just being published this week, but relying on the data that were acquired in the last 2 summers, Luxembourg, Germany, Austria, Slovakia, or Moldova. All these countries have not been considered as very much endemic for these sandflies. Nevertheless, the more we investigate the sandfly fauna in these regions, the more we actually see sometimes small, but definitely well-established sandfly populations. This definitely means that with the ongoing climatic change, we can actually consider these areas being as source areas for the emergence of sandflies into other regions that become climatically and environmentally suitable. Now, if we consider the biology of sandflies, we can point out some important differences from the mosquito and the level of the life cycle. The sandflies having the larva development in terrestrial habitats rather than the aquatic environment where the mosquito larvae do develop. And for the overwintering of the sandflies in most of the European countries, the larva stages are actually the overwintering stage. This, of course, makes them, the sandflies as the insects, very susceptible to different changes of the climate. Climatic parameters being some of the limiting environmental parameters of the sandflies' presence and survival in the given geographical regions. Therefore, we consider sandflies as being actually a very suitable indicator of how the local or global climatic and environmental changes may affect the distribution, the geographical distribution of the species, and eventually, of course, also the infection incidence and spread of the transmitted diseases that the sandflies do vector. So this is about the background of what was the driving force of establishing the CLIMOS as a project that would like to support some sort of a backbone knowledge about the sandfly emergence and presence, as well as provide some early warning system to actually point out the potential risk of the exposure of people to sandflies in different geographical areas, to give them an idea about the potential risk of sandfly-borne diseases. And therefore, we established the CLIMOS project, which comprises 29 partners from different European countries that you can see here in the overview of the project partners on the map, kindly and efficiently coordinated by Dr. Kolářová, who happens to be in the audience as well. So I'm very happy to have her here today. And we set a number of different objectives of our project that you can see here. It is a European or mostly European project spanning in the region of the endemic as well as non-endemic or not highly endemic countries for the sandfly presence. And we would like to provide data from the field to understand what is the current presence or distribution of the sandflies, what is the presence of the sandfly-borne pathogens, and use these data to actually model the potential risk of the sandfly exposure and provide some early warning system for the decision makers to support correct policies towards the sandfly-borne diseases. Now, if we consider the data so far available about the sandfly presence in Europe, what usually comes to people's mind, to those who try to seek for the maps of presence of sandflies, are these ECDC sandfly distribution maps. European Center for Disease Control has been providing these detailed maps going up to, or rather down to, the NAT3 levels of the administrative units for more than a decade now, not only for the sandflies, but also for other vectors, namely mosquitoes, ticks, and culicoides biting midges. Now, these maps, however, do have certain limitations, usually relying both on the active field surveillance as well, and actually to a larger extent, on the literature review of the data provided by other researchers. So it doesn't really actually give us the actual picture of the sandfly distribution in a given time, but rather a summary of what has been recorded in the past about different sandfly species presence, which can be very much up to date, or depending on the region, it can be heavily outdated, because as a matter of fact, in most of the European countries, there is not an ongoing frequent sandfly surveillance or monitoring system put in place for most of the time. This map actually depicts the areas where the surveillance of different types, relying on a number of different trapping techniques, is or is not done during the last decade. And you can see that actually we cannot really say that the European countries would be very thoroughly surveyed for the sandfly presence. It's mostly incidental entomological surveys rather than any frequent and long-term ongoing surveillance entomological programs. So we inevitably had to actually set up our own field surveillance, and we decided to monitor the sandfly presence in 11 countries of the project participants, stretching from Portugal on the west part of the Mediterranean up to Turkey and Israel on the east part of the Mediterranean. And we were surveying sandflies at a number of localities in each of the countries, trying to capture the actual sandfly presence, and not only that, but also their seasonality, making the season-long trappings during the period of April to November in each of the countries in a very standardized manner, because that is also very important to emphasize. Most of the data that we have from the past, from different ongoing or past projects, they have a very varying degree of standardization in terms of the trapping technique mostly, as well as the sample processing and other parameters. So we established a very firm and standardized protocol for all of the participating countries, which I think is a very unique effort, because you see very little of such large-scale surveillance in any insects, not only sandflies. And it's super important to actually collect the data that are standardized as much as possible. And we were collecting not only the sandflies, but we were also collecting some climatic and environmental parameters of our trapping sites. And also we are screening the sandflies for the presence of the sandfly-borne pathogens, which I was already mentioning at the beginning of my talk, namely Leishmania and phleboviruses. And I can only briefly introduce you into the results of this sampling effort, which we were performing for 2 consecutive years in 2023 and 2024. And we ended up with a large amount of data, which I think is super important for the other parts of our project that my colleagues will be talking about. In the first trapping season, we collected over 35,000 sandfly specimens in different countries, identifying 19 sandfly species. And you can see an overview here in the of the different participating partners in different countries, and the proportion of the trapping effort on one side and the success of the trapping on the other side. I feel a little bit humble and uncomfortable saying that actually the trapping of sandflies in the Czech Republic, as the only of the 11 so far monitored countries, ended up with a negative result. We didn't so far confirm the presence of sandflies in the Czech Republic, which we nevertheless do expect in the next decade due to the ongoing climatic changes and also due to the observance of the established sandfly populations in the nearby regions, namely in Austria, where our Austrian colleagues were actually tremendously monitoring the small, nevertheless stable and very important sandfly populations in their country. And of course, you can see from the table that some of the countries in the endemic areas of the Mediterranean region, they collected vast amounts of specimens, of which all now are under the identification, species identification. They are screened for the sandfly-borne pathogens presence. And all in all, this will be merged with the data coming from the next season, 2024, that has just been completed in autumn of the last year. And this will provide a gross bulk of data, of sandfly data, that will in a very standardized and unified manner provide the backbone for the potential modeling of sandfly presence and emergence that my colleague Suzana Blesić will be talking about in the next part of our today's webinar. We also gained some very interesting and important data about the seasonality. This is another aspect that is very unique in our CLIMOS project, because by following the presence of sandflies during the whole of the season, not only focusing on the absence or presence in one particular given sampling time point, we actually got a very good and detailed idea about the seasonality of different species in different countries. And you can see that we usually ended up with either monomodal or bimodal seasonality pattern of different sandfly species, sometimes both of the models occurring in different regions of one sampled country, as you can see in on the example of Italy, where some of the mainland Italian sampling sites were providing the monomodal seasonality of sandfly species present there, while the others, like the localities in Sicily, they actually had a different seasonal pattern. So this is again something very, very unique in terms of the seasonality data that we are collecting, that will give us a much better insight, much more detailed insight, not only about the sandfly species presence or occurrence, but actually it will give us a detailed idea about when during the season do different species occur. And by that, I would close the part of my talk, and I will be happy to pass the word to my colleagues.
Thank you very much. This was a very nice presentation. with a lot of data, and I really can't wait for the final results, as you all know. Okay, but we go forward. Please, from the audience, feel free to put your questions in the chat. We will get to them later. And then at the end, we will ask you to raise your hand and put your question in person. So don't be shy, and feel free to ask questions. These people know everything, so go ahead. And now we have Eva Kolářová to tell us more about the project. She also comes from she's a doctor, and she comes from Charles University. Her research is in the field of Leishmania, sandfly host immune interactions, and Leishmaniasis epidemiology. She has an international project lectures widely, and she co-authored the award-winning Of Parasites and Men. So congratulations on that. And Eva, please be so kind to share a presentation. Okay, not yet there.
Okay. Not yet?
Not yet.
Really? Yes.
Okay, that's it.
Okay. Good morning, ladies and gentlemen. Thank you for involving me in this. Sorry, did I change the slide now? Is everything
Yes,
okay?
you did. Yes, it changed.
Okay.
It's the blue one now.
So once more, good morning. Thank you for inviting me to participate in this Validate seminar series. And I would like to dedicate my part of this seminar to sandfly salivary proteins in leishmaniasis control, with a focus on the CLIMOS project. Sandflies salivate into the host skin when they feed on blood. The main function of sandfly saliva is to facilitate the blood intake, and by inhibiting hemostatic responses, such as coagulation and vasoconstriction. At the same time, sandfly saliva is also anti-inflammatory and modifies the site of parasite inoculation, serving as a virulence factor. In the presence of sandfly saliva, Leishmania infection could be established from a lower infective dose with higher pathology, and the lesion is more destructive and contain larger number of parasites. And this is a unique feature of sandfly saliva. This enhancing effect hasn't been observed with saliva from mosquitoes, kissing bugs, or ticks. Sandfly saliva is also antigenic, and in repeatedly bitten host, it elicits antibody production and an anti-saliva cellular immune response. Both are species-specific, and both are specific to sandfly and host species. The cell-mediated immunity has been shown to protect against severe leishmaniasis. When leishmania is inoculated into the ear of a mouse, along with sandfly saliva, it has an enhancing effect in a saliva-naive mouse. However, mice pre-exposed to sandfly saliva were protected. This concept has been applied in several studies aimed at developing a transmission-blocking vaccine. One sandfly saliva protein called PDSP15 showed quite promising results by reducing the pathology of infection and also the number of parasites, not only in a mouse model, but also in non-human primates. And the trend now is to develop vaccines that combine antigens from both the vector saliva and Leishmania. Repeated exposure to sandfly saliva also induces the production of specific antibodies. These do not protect against Leishmania infection, but could be employed as a marker of exposure and thus as a risk marker of pathogen transmission, or as to measure the effectiveness of vector control measures. And there comes the CLIMOS project, where we aim to develop a serological screening tool to assess the risk of pathogens transmission and add information for the early warning system. Such screening tool is usually based on the in-house ELISA assay that requires sandfly salivary gland homogenate as the antigen. However, there are several obstacles to dissecting sandfly salivary glands if you are planning large-scale studies. And for that, you need a sandfly colony and a person well-trained in salivary gland dissection. You can watch later this tutorial to see how delicate and time-consuming the process could be. So to overcome these obstacles, it would be better to have a recombinant antigen that represents well the whole saliva. And this recombinant protein will also have other advantages, like the standardization of the method, the scalability. It is also more economical and have lower batch-to-batch variability. And you do not need to maintain a sandfly colony. Within CLIMOS, we aim to measure exposure to sandfly bites in sentinel dog populations from Spain, Portugal, Italy, and Turkey. Our target species are therefore the main European vectors of visceral leishmaniasis, Phlebotomus perniciosus and Phlebotomus tobii, complemented by the vector of cutaneous leishmaniasis, Phlebotomus papatazi. Our workflow includes collecting a representative number of dog sera from an area with a high abundance of the desired sandfly species. Then we have tested them against sandfly salivary gland homogenate to select positive sera for the main antigen characterization in an immunoprecipitation assay complemented with Western blot. 4 selected candidate proteins were then produced in E. coli expression system. And this is the result. In previous projects, we have already identified the Phlebotomus perniciosus protein named SP3B as a reliable marker of exposure and validated this protein in several studies involving dog sera. Within the CLIMOS project, the ELISA test has been optimized, being now more sensitive and available for epidemiological studies if you are interested. For other 2 sandfly species, we tested the selected antigens against the salivary gland homogenate as a gold standard, and the results are quite promising, with at least 2 proteins of Phlebotomus tobii representing well the whole salivary antigens. Similarly, we evaluated also 2 proteins from Phlebotomus papatazi as a promising candidates for such a screening serological test. And work is in progress now to validate those proteins in a larger scale and test them for sensitivity and specificity. So I would like to thank you to my collaborators for this particular task within the whole CLIMOS project, being from Charles University in the Czech Republic, and my dear colleagues from Turkey that collected the sera. Without them, the work will not be possible. And I would like to also thank to our dear coordinator, Karla Maya, for her personal touch on this project. Thank you very much for your attention.
Thank you, Eva. This was Very, very nice. And we definitely need you in Validate Network, so please consider it based on what you told us. Yes. Okay, we have 2 questions so far, but let's go with Suzana's presentation and then we will go back to the question. Is that okay?
Okay.
Suzana, you're ready, right?
Just let me
Yes, I am.
realize how to stop sharing.
Great, you
Yes.
realized it.
Perfect.
Yes. Okay, so we have our next speaker, Dr. Suzana Blesić. She's an associate professor at the University of Belgrade, and she's leading research on biomechanics, biomedical engineering, and complex systems. With a background in statistical physics, she has contributed to major EU-funded projects on climate change and health. I was amazed when I heard from her about the CLIMOS project, and then I was even more amazed when I heard that she's a physicist. So please pay attention what a physicist can tell you about phlebotomia and leishmania. Suzana, the floor is yours.
Thank you so much. Actually, I wanted to start with me. I mean, this is not my first time, of course, that I'm listening to my colleagues from the project, and I'm always in awe, like with what they are doing and with their results. And one moment I stop, being thinking that I'm a presenter and just start being a person from the public. So, I mean, thank you also for being interested for what I have to say. But first, I'm going to tell you about what are we going to do with this data that are being collected and analyzed by people like Vit and Eva and all the others that all the others in the project. But I first have to say, and every time I have to say, that if it is not for their work and also for not for the data that they are providing to us, to the people that we within the project or data analysts and modelers, and then we will never be able to understand and monitor climate and environmental drivers of sandfly-borne diseases, and also we will not be able to produce what we call early warning systems, but they are, in the case of the CLIMOS project, there are early warning systems that show climate environmental suitability for the presence of sandflies and how this suitability is going to change with the climate change and with environmental degradation. So I'm going to just, tell you a bit what are we doing in that respect and how we are going to do that, because we are already just mostly with the preliminary results. And this is, the main output of the project is going to be this what we call within the project the model. So this understanding of climate environmental drivers of the presence of sandflies and sandfly-borne diseases, and then as the ultimate product of the project, along with all this science, wonderful science and knowledge that will come out of it as the ultimate product, is something that should be and we hope will be used. And this is the early warning system and this understanding which should help majorly health systems, but also general population prepare or be aware of the presence of the sandflies in our environments. So again, everyone was talking about this project, so me again. So now you see us. This is us. So you heard about us. It is a very, very big project. It has 29 partners from 16 countries. We started, when we started, we were already 100+ researchers, and now the network is much, much bigger. And the thing that I love about this network, but also the thing that why we got the funding, is not just that we are really, really nice people, but that this network is actually an interdisciplinary, multi-sectoral network of people doing different kinds of things. And the people who have to come together in order to tackle the problems of climate and health, and problem of the spread of sandfly-borne diseases is one of these problems. So I am the work package leader of the work package 3 of the CLIMOS project, and this is it's called data analysis and modeling and early warning system development. And here you have the picture is, I mean, something that came up at the beginning of the project. Is just a mock-up of how the early warning system will look like. So the colors are here just to see, I mean, what colors can be there, but not, they're not still showing anything, any real results. But I mean, this is, you know, what our early warning system will be. It will be a map that will show you, but in the dynamic manner, the present status, and then the projections of the climatic suitability and environmental suitability for different species of sandflies. So in order to do that, and I will tell you a bit more about each of these components, we will use and then we'll produce open data, which will be the sandfly data, so you know, past and present, so the historical data that we have collected and to some extent also systematized and harmonized, and then the data from the two sandfly monitoring seasons that we were having within the CLIMOS project. So there will be open data from these two seasons that Vít was talking about, and then all the data again for pathogens and for the diseases that we have collected, and again to some extent standardized and harmonized. So these are, again, for the pathogens, these are historical records and records from our monitoring seasons, and from the diseases we are trying to collect data about human and animal cases, mostly dog cases, from some of the endemic countries, and then all the climate, environmental, and socioeconomic records that we are going to use. We are using generally three modeling approaches to modeling One Health and Climate, or climate drivers of sandfly-borne diseases, and these are environmental niche and species distribution modeling, then a couple of approaches to machine learning and AI use in modeling, and then the time series analysis, and this is something that I do, and our colleagues from the University of Haifa. And then what we are aiming for are three time horizon forecasts for the sandfly risk, and these are not the projections, but the present status. So it is like in weather forecast, how the situation is within the few days at the moment where you look at this information, and then the forecast for the next couple of months, which is just mostly this is a seasonal forecast, and then projections with the climate change scenarios. So in the next 10 to 70 years. And then at the end, I'm just going to talk really shortly about the sandfly-borne diseases ontologies. This is something that we are also within this work package, and also within this group of data modelers and technology platform developers that we are developing within the project. So the data that we are using, so again, our main aim is to model climate and environmental drivers of the sandfly presence, and then risk of the sandfly-borne diseases. And for that, we need two main, actually three, but two main sources of data, along with data from the sandfly monitoring. One are the climate data, and here we are using, thanks to wonderful work of climatologists in the last mostly 40 years, we are using, like everyone else, the open source data sets that are available to us, different open source data sets, and I'm just showing the example of one. So this is the ERA 5 land data on different sorts of climate variables, and then in the second column you can see the breadth, the load of the climate variables that we can use in this data set, and then the third column is showing the measuring units in which these variables are given, and then these variables are given. This is a very long time series from 1950. It starts in 1950. It is an hourly series, so it has a lot of data. And then, so it has a wonderful temporal coverage, and then it has a very good spatial coverage. The spatial coverage is 10 kilometers by 10 kilometers, but within the CLIMOS, we are also going to, and this is one of the, also big things that we are going to do. Some of our partners are going, still working on it, are going to downscale this open data set to 1 kilometer by 1 kilometer coverage. So we will have also possibility to work on a very good spatial coverage with of climate data and sandfly data. The second big data set that we are using is also data set that is produced by our partner. This is this partner is UFZ. It's Helmholtz Institute for Environmental Research from Leipzig, and they're producing the soil moisture data. They have a model that is called MAJ model, which is being forced also by one open climate data set, EOPS, and they're providing daily soil moisture data for the whole Europe, actually for the whole world. If you need that, I mean, this is the source where you should go. And what they're providing for us is, again, 1 kilometer by 1 kilometer coverage with 6 layers of soil in depth. So we have like a very good in-depth information, and also, again, quite nicely focused spatial coverage, and this spatial coverage goes together with the spatial coverage that we are having with climate data. So we can work, if we are interested, the lowest cell that we can look into is a cell of 1 kilometer by 1 kilometer anywhere in Europe. Then, in order to understand what we do, in order to understand the climate and environmental drivers of the sandfly presence and then risk of sandfly-borne diseases, what we are doing, we are using this climate data and this environmental data and then the sandfly surveillance data that we are having from our partners, and that is coming again from the project and also from the historical records. And mostly this is also, I mean, their wonderful work that they were doing before. And what we are trying to do, we're trying, I mean, to model and to analyze and to make what you will call a regression model of the connections between climate, environmental, and sandfly variables. And then when we have that, then we can use this model as an input to the early warning system, as input to this map that I showed at the beginning. And these are our models, or these are our approaches. So the first one and the last one, our approaches are actually both their machine learning approaches, but the species distribution modeling is just, I mean, having in their approach much more variables that they are using as an input to their approach. They're both temporal and spatial. They're looking at the present substance of the different species of sandflies, and they look into different drivers, so like different variables that they're correlating with the sandfly data. They're using both the bioclimate variables, which are actually also the variables that are like coming from the climate variables, but are being specially prepared for the analysis of the correlations with the sandflies. Because, for example, from the climate sources, you can get, let's say, I mean, an average temperature, but then you're, I mean, in this case, people are mostly interested in actually evening or night temperature, or like minimum or maximum temperature, and this is something that you have to prepare to get from the data that you are having. So this is the, again, this is why I'm talking that some of the data sets that we are using are still being, even if they're open, they're still being pre-prepared, or like, I mean, for this kind of analysis. And these are then data sets that we are going to open when we finish for the project. So this is the bioclimate data set, and then species distribution modeling is also using land use, topography, lithography, and also socioeconomic variables like human population. And from these two approaches, we already have a climate change scenario. So they're looking like in this third, the longest projection that is going with the climate change. And then we have 3 approaches that is looking into the short term, so the deterministic, or in present or in this time, and then seasonal projections. And this is the time series analysis, as I told you, that is looking into actually the temporal correlations, and then is modeling the number of sandflies, and it's looking just in meteorology, hydrology, so in just these two big data sets that I showed you. And then FIPO, this is the model that is developed within the project, and this is also a machine learning model that is looking into temporal and spatial modeling of the abundance of sandflies, and looking, as you can see, like every machine learning model, into a lot of different variables, so meteorology, land cover, hydrology, and topography, which already has some preliminary results for the short-term projections, and then seasonality modeling that is being done, again, as a time series analysis on the temporal level. And this is also, again, looking into meteorology, and it is having some preliminary results at this moment. So at this moment, we have sort of cracked down the climate projection part of our modeling and are still working on the short-term part of our modeling. And this is because, I mean, this is how a project works, but also I have to again come back to what Vit said, that because the sandflies are much more complicated for modeling than mosquitoes, so there is also a difference in there. And while most of us that came to this modeling part by knowing, you know, or having some sort of experience with mosquito modeling, are now facing like much bigger challenge with the sandflies. And at the end, I'm just going to tell you about the ontologies that we are developing. So we are working on developing, this is like a metadata language, or the machine language that is connected to sandfly-borne diseases. So we will introduce actually a new language, machine language, for the sandflies and sandfly-borne diseases that will represent data collected and published in CLIMOS, and this will be about sandfly species, parasite species, trap properties, and observation properties. And then when we do that, we will link the terms that we are introducing, and this is the terms that we are working on now. We will link these terms to other vocabularies and ontologies that are already present, and there are up to vocabularies and ontologies about different vectors or different vector-borne diseases. And then at the end, this is going to be published on the OGC Rainbow cloud. OGC is one of our partners, and this data will be also open, and this catalog will be then like a part of the machine recognition of these words that everyone is using after the end of the CLIMOS project. And that's it. Thank you so much.
Wow, that was a lot.
Sorry.
Thank you. No. That was a lot of information and a lot of new stuff that you guys are going to present. So the future will be different once you finish your project. Okay, we have a few questions in the chat. I think we can start with that, unless Sabella, Lison, and Wesleti would like to pose these questions out loud. Okay, I will start reading the first one from Sabella. Are you able to establish the months where there is high prevalence of the sandfly?
Yeah.
Bit probably for you. Yeah.
I think I can pick up this one. Yeah, thank you very much for the question. Indeed, yeah. Well, this is actually one of the aims of the so orchestrated effort in the field for 2 consecutive years, because this is indeed what we would like to aim to understand the seasonality of different species throughout the whole season, which is markedly different from what many of the other mapping projects gathering sandfly data presence have been doing so far, including ECDC efforts and others. We would like to understand not only the presence, but also the abundance and the climatic and environmental drivers to indeed, yes, be able to say which are the high season months for the sandflies in different regions of Europe, which apparently differs for the geographic regions as well as the species. So yes, this is one of our aims.
Great, thank you. The next is not a question, is actually a comment. So Lison said, thanks, the video for sandfly SG dissection is so informative, it is very tricky. Would you like to comment on that? I also know it's tricky because it's tiny and everything is so fragile and you have to be very careful, but yeah, okay.
It was quite a challenge to make this video. I have to learn some other techniques, how to mount the mobile phone to the dissecting microscope and so on. But it was a nice job to prepare this one.
Yeah, I can imagine.
They like it.
I can imagine you with all the technique trying to record it. Very good. You have the invitation. Since you're here, you must be interested in Leishmania and phlebotomine, and you can join Validate with the link. There is a link in the chat if you would like, and if you're not already a member. And then we have another question for Vit. You mentioned that the subgenus Larusis is the predominant one. The question is as follows. Phlebotomina identification was based on morphology, molecular study, multitophs, or both? And did you identify males and females?
Yeah, great. Hi, Barbara, how are you doing? Yeah, indeed, this is also very good question. And yes, we actually found Larusius species to be predominant in most of the surveyed countries. That's true, not all of them though. Nevertheless, the species identification was actually done by a combination, by an integrative approach using the morphology as well as molecular techniques. Multitoph protein profiling has also been one of the methods of choice, and of course, sequencing DNA barcoding mainly was also heavily deployed. But luckily, also to say, among the CLIMOS consortium, many of the partners are actually prolific and very well experienced entomologists. So that's a great asset of our consortium, to say, and so they are able to deploy a combination of different techniques, and we have a standardized SOP focusing on that. So and yes, males and females were identified. We distinguished for the particular situations at different trapping sites, outlining the different approaches depending on the number of the specimens caught at one site. So if it is a huge catch, we identify a representative proportion. However, in most of the sites, people were actually identifying all the specimens, males and females alike. And in the females, of course, different approaches were deployed, also with bearing in mind that the pathogen screening will be performed on those. So we have outlined some standardized approach for that as well.
Thank you, Vit. Now we have one for Suzana. Thank you for this informative presentation. Questions to the last speaker. You said sandflies are more complicated to monitor than mosquitoes. Could you please explain why? So, Suzana.
Yeah, they are to model. Well, because I mean, first the biology and then I mean the way how I mean the cycle of development is going is not that well known, and because they're like they're super tiny and super sensitive to everything. I mean, this cycle can change with like, I mean, really microclimate changes. And this is something that is really complicated to model because as you saw, our like filter, I mean, the best filter that we have is 1 kilometer by 1 kilometer of data. So we cannot like look into the microclimate. We need to like put it bigger. So we have to generalize something that is like really small into this cell. Even if we are having also the microclimate data, so we are looking into that. So this is the first thing. And then the second thing, what is really complicated is because we are looking in the fly-borne diseases risk, is the cycle of infection that first it goes through the host, but then because especially in Leishmania, it doesn't like in mosquito-borne diseases, you're not like being bitten by the vector, and then after some time you develop a disease. But this disease, I mean, the Leishmania you can develop it anytime between like, you know, now to several years. So so that actually cannot be easily modeled. And because we have that this complication, then we have to look into the data, and this is what we are doing in order to sort of have the better picture. We are looking into the data from the sandfly monitoring, and then we look into the animal and human cases data in order to have these three, levels of transmission of disease in order to understand as much as we can the risk of appearance of the sandfly-borne diseases if you have sandflies on the ground.
Thank you, Suzana. Feel free to raise your hand. I think do we have a few more minutes, Blake? Okay, we have Lison.
Hi. Can you hear
Hi.
me? Yeah.
Yeah.
Hello. Thank you for all the presentation. It was very informative. I just have a question. I know it's very difficult, but would you try to collect some immature stages of sandflies also to get some information on the breeding sites or try to target them by vector control later?
Yeah, I think I can pick up this one as well. Hi, Lison, how are you doing? I see you.
Hi.
Well, yeah, actually, we decided not to focus on the immature stages, even though actually that would be also super interesting because, and you really touch a very, very good point here because when Suzana was talking about how the sandflies may be different in their biology from the mosquitoes, this is one of the particle hotspots or rather gaps of knowledge at the same time in what we know about their biology and their actual natural occurrence, because the breeding sites in nature, while they are very well defined for the mosquitoes, I would say, and I really envy my mosquito colleagues with that because they can very, very efficiently pinpoint the breeding sites in the environment, in the countryside, depending on the habitat. We know very little about that in sandflies. So this is unfortunately beyond the scope of the CLIMOS project, but definitely something to pursue in the future. And we are actually having another project with one of the CLIMOS partners, namely our Austrian colleagues from the Medical University of Vienna. I think Kati Platzgummer is here in the audience, and maybe others as well. So we are trying to target the breeding sites of Phlebotomus massiti, which is the species that does occur in Austria. Not hyper-endemic sandfly country, nevertheless the one with already well monitored and healthy sandfly population, even though small and limited. A short answer would be no, but we regret it because it would also be a great additional super valuable information. So this is something that definitely needs to be addressed in more details in the future research.
Okay, thank you. Yes, that would be very interesting. Thanks.
Thank you.
Hi, Sarah, it's Blakely. I'm just going to let you know we officially have till 12:30, but that is obviously if the speakers are happy. So there's no worry on the time.
Okay, great. Because we have one more comment question in the chat, and then we have Olenka wanting to speak. So let's go to Soraya's question. Again, for Vit, you are the champion today. You know, everybody's asking you. Thanks for the presentation. Did you check the sandfly infection by Leishmania parasite?
Not the champion, maybe my presentation was the clumsiest of all three because people keep asking me, but nevertheless, thank you for the question. Of course, yes. Actually, that is one of the aims of CLIMOS as well, not only to understand the sandfly species composition and geographical distribution, but we indeed also screen them for not only for Leishmania, but also for the flaviviruses. So yes, this will be another of the important outcomes, because unlike the focal limited studies in particular transmission sites or transmission cycle areas, this will again give us an overall understanding of what is the current Leishmania and flaviviruses presence or occurrence in the countries within the sampling of the CLIMOS project. So yes, we still cannot present much of the results on that since it is an ongoing process. But at the end of the project, we will be having the results of the screening of Leishmania and flaviviruses. Yes.
Fantastic. And then we have one for Dr. Iva Kolářová. Thanks for the presentation. And the question is, what about the co-localization of different species and sandfly?
If I understood well, it's about like a sympatrically occurring sandflies and the specificity of the test. Is that correct?
Exactly. That's it.
So, the antibody response is quite specific. So you can distinguish between different vectors, but this should be tested more using well-defined sera, which are not so much available, so it's quite tricky to test this species specificity of the test. But based on the mouse model, the antibody response is quite specific, and there shouldn't be a cross-reactivity between the antigens of Phlebotomus perniciosus and Phlebotomus papatazi. So, but the tests are ongoing in our lab, and I will tell you more later during this year.
Thanks a lot.
And what I can add more concerning the two vector species of Leishmania infantum, Phlebotomus perniciosus and Phlebotomus tobii, they are closely related, and there is a possibility of cross-reactivity between the antigens, but those species are so far have different geographical distributions. So there is no like a need to distinguish between those two.
Okay, thank you. Thank you very much. We have a comment from Alessandro, who is very curious to see and try the EWS platform as well as I am. So we can't wait. And we go now to Alenka. Sorry for waiting. Thank you for wanting to pose your question in person. So can you ask your question, please?
All right, thank you very much. Sorry, I don't know if this has been asked or has been said because I have got network connectivity. But my question is, I live in Nigeria, which is on the continent of Africa. One thing I couldn't pick from the presentation is in terms of the ecology of the leishmaniasis parasites or the insect themselves. Like, what is the ecology like? General across some of the studies that has been done. Also, in terms of the process that you go about capturing the insects, how do you go about it? What is your setup like? I would be interested in knowing that. Thank you very much.
Bit, can I just take over? I mean, I would just start with ecology and then if you can go with it. So that, I mean, from the modeling point of view, thank you for that question. We are having great and complicated communications and discussions within the project about that, because this is the most complicated thing. And it's actually, I mean, the question that is coming from you is the questions that we are having, not the questions, but interventions that we are having from our mostly Israeli colleagues, because they also have sandflies. They're an endemic endemic place, but like with completely different ecology. And this is still something about which we are discussing within our modelers group. So we also don't we are also struggling with that. And of course, I mean, you know, our models will come up with, I mean, what we have from our data, and mostly, I mean, we will have these ecological niches that will show the ecological suitability for some or different species of sandflies. But I think that this question is going to be open after CLIMOS still.
Yeah, I completely agree on that. I think this is a bit of a pioneering project in terms of developing such a warning system, but the actual ecological requirements of sandfly species in Europe considerably differ from the sandflies occurring in other geographical regions, being in sub-Saharan Africa or Southeast Asia or other parts where these sandflies do occur. So I think we're kind of putting down a concept that maybe sort of universal, but at the same time, we definitely need to think very carefully when trying to adapt the system on the geographically very different areas, because they are indeed very different in the ecology of sandfly species occurring locally, as well as the dynamics of the Leishmania transmission. We can see that clearly on the example of regions that are not so far from our study area, but already in North Africa, in the Maghreb countries, we see that the ecological traits of the sandfly species occurring there there are quite different, and we suppose that also the dynamics of the transmission, and we know that the dynamics of the leishmaniasis transmission there are quite different. So I mean, this is a thing that kind of can be probably taken and very carefully adapted to local conditions rather than being very, very universal. I hope I replied to the question.
Thank you. Are you happy with the answer?
Yes, I am. I think, I don't know, the last one is on the how to capture the insects. What is the setup like? How are they able to trap the insects?
Right. Yeah. We really put a lot of emphasis on the standardization of the trapping method. So among the methods that are available, we simply focused on the CTC light traps, which is a universal trapping method that has its limitations. We are aware of that, but generally speaking, it seems to be from different points of view, taking into account the logistics as well as the natural conditions in different areas. It seems to be the best fitting trapping method for our purpose among the whole of the CLIMOS geographical coverage. So is the CTC light traps that we are largely using.
Okay, thank you. I don't see any hands up, and I don't see any more questions in the chat, but I would like to ask you, all three have mentioned the chairperson of your project, and she's here, and I would ask Carla Maya, would you like to say something? Oh, there she is.
I'm here. Good morning, everybody. Thank you so much for this opportunity. Congratulations to all the presenters. I knew that you will nail it. Thank you so much for presenting the first results, to explain our project and also to present the first results. We are all very excited about it, and hopefully we will have more data to share with you soon. So please keep following us, and tomorrow we have another webinar. So, and thank you so much to validate this opportunity to share our work with so many colleagues from different countries. And as Vit was mentioning, maybe we can transpose some of our results or our work to other parts of the world. Of course, they need to be adapted, but yeah, it's, as Vit was mentioning, this is a pioneering work. It's a pilot one, so let's see how it goes. Thank you so much, Sarah.
Thank you, Carla. I have a feeling that this is not your last webinar in Validate. And so when you have the final results, I have a feeling that we will have more of you. But I would probably turn to Blakely.
Yeah, hi, everybody. I just want to say thank you to our amazing chair and our speakers. And if everybody would like to show their appreciation, probably by just typing a note in the chat, or you can turn on your camera and wave and applaud, something like that. But I think I'm going to stop the recording if that's okay, Sarah. Great.